So hi again. Now we're going to talk about protein-mediated ion transport in mitochondria, and I'm going to start by talking about metabolite transport mainly. So a thing to remember about protein-mediated ion transport is that the properties of transporters or carriers that take substances across membranes are actually very similar to the properties of enzymes. So you can really think of this transport as you think about catalysis. So similarly to enzymes, transporters in mitochondrial membranes are going to be stereospecific. They're going to recognize D versus L. They're going to recognize spatial characteristics of the substrate. They can be regulated by all the mechanisms that regulate enzymes. And also they require the expression of specific proteins. And therefore, you're going to have tissue and organelle-specific expression of these proteins and also characteristics of transport. So you know that it's a protein-mediated transport when you can inhibit it and it's different in different tissues, and when you can remove it by removing a specific gene, for example. Interestingly, in mitochondria, there's the inner mitochondrial membrane potential. And the inner mitochondrial membrane potential is really a central part of metabolite transport. Most of protein-mediated transport is going to use this membrane potential as a driving force to accumulate different metabolites in mitochondria. So you're going to see a lot of symport of these metabolites with protons, so that the proton goes down the proton gradient. You can see antiport with protons in the case of substances that are transported out of mitochondria. And you're also going to see electrogenic import, in which the net result is bringing a positive charge into the mitochondrial matrix. And that's going to be favorable because of the negative characteristic of the mitochondrial matrix normally. Many members of protein-mediated ion transport are members of the SLC25A family. So this is a large family of transporters. They seem to have derived from an original transporter that transported anions, and they transport all sorts of different things in mitochondria. They transport amino acids, nucleotides, cofactors, even some inorganic molecules like iron and phosphate, and also many different substrates into mitochondria are transported by members of this family. And they all have similarities in their structure. They all seem to have evolved from an original anionic transporter in mitochondria. So an example of a member of this family that's the most abundant protein actually in the inner mitochondrial membrane is the ANT, or the Adenine Nucleotide Translocator. This protein is responsible for the exchange of ADP for ATP in the inner mitochondrial membrane. The result of the exchange of ADP with 3 negative charges for ATP with 4 negative charges is the net uptake of a positive charge. It's equivalent to throwing a negative charge into the intermembrane space or taking up a positive charge into the matrix. So this is an electrogenic import, and therefore it's favorable in the presence of the inner mitochondrial membrane potential. Obviously, it's a very necessary transport because the ATP synthase uses ADP to produce ATP inside mitochondria, and most ATP in the cell is going to be used outside mitochondria, so it has to be transported out of the inner mitochondrial membrane. The mechanism for this protein is known because this protein has already been crystallized, so we understand its structure very well. We know that it can face the outer side of the inner mitochondrial membrane, so the intermembrane space side of the inner mitochondrial membrane, and here it binds ADP. This binding changes the structure of this protein and makes it now face the matrix side of the membrane and also release ADP. The protein now has an affinity for ATP. It will bind ATP, change its structure again, and now face the intermembrane space once more and release ATP into the intermembrane space, into the outer side of the inner mitochondrial membrane. So it cycles between binding and transporting ADP into mitochondria and binding and transporting ATP out of mitochondria. Because it has these different structures, it actually has different inhibitors. So you can freeze this protein into the configuration in which it faces the outside by treating mitochondria with atractylazide or carboxyatractylazide, which is a non-competitive form of this inhibitor. This inhibits transport and also puts the protein into the specific configuration facing the outside. You can also freeze the structure with its ATP binding structure facing the mitochondrial matrix by treating mitochondria with buncrenic acid or buncrocate. So you know that there are two different structures, and both of these structures can be inhibited quite specifically with these natural molecules. This gave us a sense that this transport initially was mediated by a protein, and today also it gives us an idea of how this protein participates in many different processes. And it's actually such an abundant protein in the inner mitochondrial membrane that it really participates even in the shape of mitochondria. So when you treat mitochondria with atractylazide or with buncrocate, you actually change light scattering properties and you change the whole shape of mitochondria. In mammals, there are 4 different isoforms of this transporter, extremely important protein. And one important characteristic is that because it exchanges ATP for ADP, it actually doesn't change the whole adenine nucleotide pool within the mitochondrial matrix. It's just exchanging one for the other. So there's a second transport mechanism that imports new ATP into mitochondria, that brings new ATP molecules. These are proteins from the CAM-C family, different protein family, much smaller expression, much less activity, but then again, you have to recover ATP quantities in mitochondria much more rarely than you have to exchange ADP for ATP. So that's the adenine nucleotide translocator, the most abundant protein in the inner mitochondrial membrane. Another really important, metabolically important protein is the phosphate transporter in mitochondria. It catalyzes typically the symport of phosphate with a proton, which is sometimes indicated based on kinetic studies as the antiport with an OH. But actually, the symport with a proton works perfectly fine in terms of understanding that this goes down also the delta pH potential in mitochondria. This is not an electrogenic transport because you're transporting a negative charge with a positive charge. It's going to be influenced, therefore, by delta pH, but not by delta psi because you don't have a difference in charge with this transport. The phosphate transporter can be inhibited. It's inhibited by thiol-binding molecules such as mersallyl or N-ethylmalamide, but these are not specific or selective inhibitors of the phosphate transporter. They just indicate that this is a protein transporter. We don't have very good molecules specifically to inhibit phosphate transport into mitochondria yet. Another really important transporter in mitochondria is the transporter for pyruvate. So pyruvate is generated through glycolysis in the cytosol, and then in a very central point in metabolism, pyruvate is transported into mitochondria, and inside mitochondria becomes acetyl-CoA, which then enters the Krebs cycle. Now, very surprisingly, we knew that there was a pathway to transport pyruvate into mitochondria, although it could possibly be protonated and diffused through the membrane. There was evidence based on kinetic studies that there was actually a protein pathway for this. But we didn't know what the molecular identity of this pathway was until 2012. And this actually is quite characteristic of mitochondrial transporters. We have really only found out the molecular identity of many of them in the last few years, and even transporters that are very central towards metabolism. And there are quite a few of these transporters whose molecular identity we still don't know. So this finding by Jared Rutter's group is very central in metabolism and is actually pretty recent. So this was a big hole in our understanding of metabolism that we learned about quite recently, what the pyruvate transporter is. The pyruvate transporter has a family of its own, so it's called the MPC family of the mitochondrial pyruvate carrier family. It's not of the SLC25A family like the other proteins we saw before. These families have 3 transmembrane alpha helixes, probably more than one subunit acting together so that they can actually transport a molecule like pyruvate. Pyruvate is thought to be co-transported with a proton, therefore down delta pH again, and it can be specifically inhibited, this transporter, by a molecule known as UK5099. So you can specifically inhibit transport of this substrate into mitochondria, even in intact cells, and determine what kind of substrates are used in mitochondria using this pharmacological tool as a poison in mitochondria, so you can see the contribution of different substrates. That's what I wanted to talk about, individual metabolite transporters. I just want to remind you that there are quite a few other metabolite transporters, and I'm not going to go through these transporters one by one, but I want to remind you that these transporters can act concertedly and generate activities of transport that are not the molecules that they transport themselves. And I think the clearest example of this are the shuttles in which electrons reducing NAD to NADH in the cytosol are transported into mitochondria without the transport of NADH itself. So this is the glycerol phosphate shuttle, in which electrons from NADH are going to be transported into the mitochondrial membrane, reducing coenzyme Q in the mitochondrial membrane by glycerol phosphate dehydrogenase, which is an enzyme that's on the outer side of the inner mitochondrial membrane. A second shuttle system is the malate aspartate system, which is basically a set of reversible reactions and reversible transporters in the inner mitochondrial membrane that concertedly is going to transport 2 electrons from NADH in the cytosol to 2 electrons on NADH in the mitochondrial matrix. And it does this by transporting and transforming glutamate, aspartate, oxaloacetate, and alpha-ketoglutarate, as well as malate, with transporters that already exist in mitochondria. So the net reaction is transport of nothing except 2 electrons, but there are lots of transformations happening for these 2 electrons to be transported into mitochondria. So these are more physiological transporters of different metabolites, transporters of organic molecules. I'm going to come back in the next video and talk to you about inorganic ion transport in mitochondria. We're going to talk about calcium and potassium transport in mitochondria. Till then.